students, we are going to discuss about organs of immune system. Based on the function, there are three types of lymphoid organ. They are primary lymphoid organ, secondary lymphoid organ, and tertiary lymphoid organ. When we look into the primary lymphoid organ, it is the sites where production, development, and maturation of the cells occurs. There are two types of primary lymphoid organs. They are bone marrow and thymus. Next, we are going to discuss about bone marrow, which is the primary lymphoid organ which is responsible for the or support the self-renewal and differentiation of hematopoietic stem cells produced through hematopoiesis, which is the active sites of hematopoiesis. There are lots of active sites such as long bones, such as femur, humerus, and hip bones such as ilius and sternum tend to active sites of hematopoiesis. Bone marrow is not only responsible for development and the replenishment of blood cells, it is also responsible for the maintaining pool of HSCs throughout the life of an adult vertebrate. When we take the cross section of a bone marrow, there is a tightly packed HSCs were tightly packed with a kind of cells called the stromal cells. Stromal cells provide a hematopoiesis inducing environment for the differentiation of hematopoietic stem cells. It is the site where origin and development of B cell take place, and it is also the site where T cell origin take place, but the maturation is take place in thymus. In birds, instead of bone marrow, they have a organ called bursa fabricus. In cattle and sheep, primary tissue hosting the maturation, proliferation, and diversification of B cell early in gestation in fetal spleen. In rabbits, proliferation and diversification of B cell. They use gut-associated tissue or GAT, such as appendix, as primary lymphoid tissue. We are going to discuss about thymus, the next primary lymphoid organ, which is the site of T cell development. The origin takes place in bone marrow. The development takes place in thymus, which is a flat bilobed organ just situated above the heart. Each lobe is surrounded by a capsule and is divided into lobules which are separated from each other by the strands called trabeculae. Each lobule again divided into two compartments such as outer compartment cortex and inner medulla. The cortex is densely packed with immature T cells called thymocytes, and inner compartment of medulla is sparsely populated with thymocytes. And both cortex and medulla is crisscrossed with the three-dimensional stromal network, which is composed of epithelial cells, dendritic cells, and macrophages, which the framework of organ provide or contribute to the growth and maturation of thymocytes. There is a kind of cells called nerve cells, which is the thymic epithelial cells in the outer cortex, which have a long membrane extensions that surround us as many as 50 thymocytes, forming a large multicellular complex. Remaining epithelial cells have interconnecting cytoplasmic extension that form a network and have been shown interaction with the numerous thymocytes. As thymocytes develop, an enormous diversity of T cell receptors is generated by a random process that produces some T cells with receptors which is capable of recognizing antigen MHC complex. Most of the T cells which are incapable of recognizing antigen MHC complex and some react with self-antigen MHC complex are eliminated through a process called apoptosis by thymus. Thymus reduces the death of those T cells which cannot recognize antigen MHC complex by process apoptosis, and thereby reduce autoimmune diseases. More than 95% of cells were destroyed by this process. As earlier mentioned, the cells which cannot recognize self and non-self were eliminated by process called apoptosis and cause a chance of autoimmune disease. Another factor which influences the thymus is aging. Aging is accompanied by decrease in thymus function. As age increases, the size of the thymus decreases with the aging. They have maximum size at puberty and it decreases with aging. There is a decrease in cortical and medullary cells and increase in total fat content of the organ at the increase in aging. Next, we're going to discuss about secondary lymphoid organ. The secondary lymphoid organ's main function is trap antigen and provide sites for mature lymphocytes to interact with that antigen. There are specialized and highly organized and less organized lymphoid tissues. The highly organized lymphoid tissues examples were lymph node and spleen, and less organized one were mucosal associated lymphoid tissues and such as gut associated lymphoid tissue are the secondary lymphoid organs. Next we discuss about the lymph node. The first secondary lymphoid organ is a highly specialized or highly organized secondary lymphoid organ, which is the sites where immune responses are mounted antigens in lymph, which is encapsulated with a bean-like structure containing reticular network, packed with lymphocyte, macrophages, and dendritic cells. Actually, lymph nodes are cluster junctions of lymph cells, where, as earlier mentioned, it is the site where immune responses are mounted to antigens. Lymph node is divided into three concentric regions: cortex, paracortex, and medulla. Cortex is the outermost layer which contains lymphocytes, mostly the B cells, and macrophages and follicular dendritic cells arranged in primary follicles. Since it contains mostly B cells, it is called a thymus independent region. After an antigenic challenge, if there is an antigenic challenge comes, a primary follicle become larger secondary follicle, a ring of concentrically packed lymphocytes surrounding a center around the center. The center is called a germinal center. Secondary follicle means a ring of concentrically packed B lymphocytes, and it has a center. It is called a germinal center, which finds a focus of proliferating B lymphocytes, an area that contains non-dividing B cells and some helper T cells interspersed with the macrophages and follicular dendritic cells. As the B cells mature and differentiate into plasma cells and produce antibodies, which destroys the antigens, and T cells, T helper cells also helps in this process for the activation of B cells, etc. Next, we are going to discuss another region, paracortex, which is situated just beneath the cortex, which is populated by T lymphocyte. So, it is called a thymus dependent region compared to cortex, where there is no lymphocyte or violet lymphocytes. It is called a thymus independent region, which is largely populated by T lymphocytes and contains interdigitating dendritic cells. The dendritic cells were migrated from tissues to the node. The interdigitating dendritic cells function is which express high levels of class 2 MHC molecule, which is necessary for the antigen presentation to TH cells. Paracortex is the region just beneath the cortex. Okay, next we going to another region called medulla, or the last region, medulla, where through the it is the region through the lymph ooze out. The lymph comes through afferent vessel and oozes out through efferent vessel. Efferent vessels were present just in the region medulla. And medulla is sparsely populated with the lymphoid lineage cells, where B lymphocytes and T lymphocytes were rarely seen, and mostly plasma cells or many plasma cells were present there. They secrete antibody molecules. When lymph comes through the afferent vessel and goes through these regions such as cortex, paracortex, medulla. In cortex, it undergo humoral immunity and or phagocytosis, and in paracortex, it undergo cell-mediated immunity, medullary humoral immunity. So, both humoral and cell-mediated immunity will take place in the lymph node, and the antigen-free lymph oozes out through the efferent vessel. Yes, we are going to discuss about the secondary organ. It is a highly organized structure which uh, mount immune responses to antigens in the bloodstream. Lymph node in the lymph and spleen concerned with the antigens in the bloodstream. Spleen is a large ovoid secondary lymphoid organ which is situated just high in the left abdominal cavity, which specializes in filtering blood and trapping bloodborne antigens. Thus, it can respond to systemic infections. And bloodborne antigens and lymphocytes are carried into the spleen through the splenic artery. There are two types of vessels, blood vessels in the spleen. They are splenic artery and a splenic. The blood from the body and cells were carried into the spleen by splenic artery and carried out by the splenic vein. The antigen-free blood is carried out by splenic vein. The spleen is surrounded by a capsule which extends into numerous projections like all other organs. They are called trabeculae. It is from a compartmentalized structure. There are compartments are two types, red pulp and white pulp, which is separated by a marginal zone. The red pulp consists of a network of sinusoids populated by macrophages and numerous red blood cells or erythrocytes and few lymphocytes. It is a site where old and defective red blood cells are destroyed and removed. So, spleen is also called a graveyard of erythrocytes. The splenic white bulb is the next region. The splenic white bulb surrounds the branches of splenic artery, forming a periarteriolar lymphoid sheath populated mainly by T lymphocytes. Primary lymphoid follicles are attached to periarteriolar lymphoid sheath. These follicles are rich in B cells and some of them contain germinal centers. The marginal zone located peripheral to the PALS is populated by lymphocytes and macrophages. Bloodborne antigens and lymphocytes enter the spleen through splenic artery, which empties into the marginal zone. In the marginal zone, antigen is trapped by interdigitating dendritic cells, which carry the antigen to PALs or periarteriolar lymphoid sheath. Lymphocytes in the blood also enter the sinuses in the marginal zone and migrate to the PALs. Upon antigenic challenge, these primary follicles develop into characteristic secondary follicles containing germinal centers, where rapidly dividing B cells and plasma cells are surrounded by dense clusters of concentrically arranged lymphocytes. When the antigenic challenge comes, the primary follicle develops into secondary follicle containing germinal cells and forms B cells which mature into plasma cells, and plasma cells produce antibodies. Next, we are going to discuss about the mucosa-associated lymphoid tissue, the less organized secondary lymphoid tissue. It is found in the mucous membranes lining the digestive, respiratory and urogenital system, major sites of entry of most pathogens. These are vulnerable membrane surfaces are defended by group of organized lymphoid tissue mentioned earlier and now collectively as mucosal-associated lymphoid tissue. These tissues range from loose, barely organized clusters of lymphoid cells in the lamina propria of intestinal villi to the well-organized structures such as foliar tonsils, appendix, as well as Peyer's patches, which are found in the submucosal layer, and its number of plasma cells is far exceeds in the spleen, lymph node, and bone marrow. Based on the position where the mucous membrane is found, they are named as GALT, NALT. If it is GALT, it is gut associated lymphoid tissue, it means found in intestine, and NALT, nasal associated lymphoid tissue, which is found in the respiratory tract of ours. Peyer's patch occur in the submucosal layer present beneath the lamina propria under the epithelial layer of intestinal villi. Each Peyer's patch is a nodule of 30 to 40 lymphoid follicles. After antigenic challenge, it turned to secondary follicle. Lamina propria occurs under the epithelial layer of villi. Populated with a larger number of, which means lamina propria is populated with larger number of plasma cell macrophages, activated T helper cells in loose clusters, M cells located in the mucous membranes, endocytos, antigen from the lumen of the digestive, respiratory, and urogenital tracts. The antigen is transported the cell and released into the large basolateral pocket. Antigen transported across the epithelial layer by M cells at an inducing site activate B cells in the underlying lymphoid follicle. The activated B cells differentiate into IgA producing plasma cells which migrate along the submucosa layer. The outer mucosal epithelium contains intraepithelial lymphocytes. The activated B cells differentiate into plasma cells which leave the follicles and secrete the IgA class of antibodies. These antibodies then are transported across the epithelial cells and release the secretory IgA into the lumen where they can interact with the antigens. As we earlier discussed about the primary lymphoid organ, secondary lymphoid organ, and it is the tertiary lymphoid organ. Tertiary lymphoid organs are the organs where lymphocytes activated by antigen in secondary lymphoid tissue can return to these organs, such as examples for lymphoid organs were lung, liver, brain. Those who reacts with the antigen in secondary lymphoid organ returns as affected cells or memory cells to the tertiary lymphoid organ. So it provide tertiary lymphoid organ provide an environment for the return of lymphoid cells. Recent researchers found that brain, for instance, established reticular systems that guides lymphocytes respond to chronic infection with the protozoan that causes toxoplasmosis. Here are the references.